Beta oxidation is the process by which fatty acids, in the form of acyl-CoA molecules, are broken down in mitochondria and peroxisomes to generate acetyl-CoA, the entry molecule for the citric acid cycle. The beta oxidation of fatty acids involves 3 stages. 1. Activation of fatty acids in the cytosol. In the cytosol of the cell, long-chain fatty acids are activated by ATP and coenzyme A, and fatty acyl-CoA is formed. Short-chain fatty acids are activated in mitochondria. The ATP is converted to AMP and pyrophosphate, P-pi, which is cleaved by pyrophosphatase to 2 inorganic phosphates, 2-pi. Because 2 high-energy phosphate bonds are cleaved, the equivalent of 2 molecules of ATP is used for fatty acid activation. 2. Transport of activated fatty acids into mitochondria, carnitine shuttle. Fatty acyl-CoA from the cytosol reacts with carnitine in the outer mitochondrial membrane, forming fatty acylcarnitine. The enzyme is carnitine acyltransferase I, CAT-I, which is also called carnitine palmitol transferase I, CPT-I. Fatty acylcarnitine passes to the inner membrane, where it reforms to fatty acyl-CoA, which enters the matrix. The second enzyme is carnitine acyltransferase II, CAT2. Carnitine acyltransferase I, which catalyzes the transfer of acyl groups from coenzyme A to carnitine, is inhibited by malonyl-CoA, an intermediate in fatty acid synthesis. Therefore, when fatty acids are being synthesized in the cytosol, malonyl-CoA inhibits their transport into mitochondria and, thus, prevents a futile cycle, synthesis followed by immediate degradation. Inside the mitochondrion, the fatty acyl-CoA undergoes beta oxidation. 3. Beta oxidation proper in the mitochondrial matrix. Beta oxidation, in which all reactions involve the beta carbon of a fatty acyl-CoA, is a spiral consisting of 4 sequential steps, the first 3 of which are similar to those in the TCA cycle between succinate and oxaloacetate. These steps are repeated until all the carbons of an even-chain fatty acyl-CoA are converted to acetyl-CoA. FAD accepts hydrogens from a fatty acyl-CoA in the first step. A double bond is produced between the alpha and beta carbons, and an enol-CoA is formed. The FADH to that is produced interacts with the electron transport chain, generating ATP. Enzyme, acyl-CoA dehydrogenase, multiple variants of this enzyme. H2O adds across the double bond, and a beta-hydroxyacyl-CoA is formed. Enzyme, enol-CoA hydratase. Beta-hydroxyacyl-CoA is oxidized by NAD+ to a beta-ketoacyl-CoA. The NAD that is produced interacts with the electron transport chain, generating ATP. Enzyme, L3-hydroxyacyl-CoA dehydrogenase, which is specific for the L isomer of the beta-hydroxyacyl-CoA. The bond between the alpha and beta carbons of the beta-ketoacyl-CoA is cleaved by a thiolase that requires coenzyme A. Acetyl-CoA is produced from the 2 carbons at the carboxyl end of the original fatty acyl-CoA, and the remaining carbons form a fatty acyl-CoA that is 2 carbons shorter than the original. Enzyme, beta-ketothiolase. The shortened fatty acyl-CoA repeats these 4 steps. Repetitions continue until all the carbons of the original fatty acyl-CoA are converted to acetyl-CoA. The overall reaction for one cycle of beta oxidation is CN-acyl-CoA plus fat plus NAD plus H2O plus CoA CN to acyl-CoA plus FADH2 plus NAD plus H plus acetyl-CoA. Regulation. Beta oxidation is regulated by the mechanisms that control oxidative phosphorylation, i.e., by the demand for ATP. Activators. Epinephrine stimulates beta oxidation by activating a CAMP-dependent protein kinase, which leads to the phosphorylation and thus activation of HSL. When activated, HSL releases fatty acids and glycerol from adipose tissue for beta oxidation. Inhibitors. Insulin inhibits beta oxidation by dephosphorylating HSL and thus inhibiting the release of fatty acids from adipose tissue. Fatty acids, which are the major source of energy in the human body, are oxidized mainly by beta oxidation.